Hi, my name is Kamille Barrientos, and our presentation is on Alzheimer's disease, causes and symptoms, and keratin structure and functions. What is Alzheimer's disease? Alzheimer's disease is a progressive neurologic disorder beginning with mild memory loss and possibly leading to loss of the ability to carry on a conversation and respond to the environment. Alzheimer's disease is the most common cause of dementia and is a continuous decline of brain functioning, behavior, and social skills. Causes of Alzheimer's disease. Scientists believe that for most people, Alzheimer's disease is caused by a combination of genetic, lifestyle, and environmental factors that affect the brain over time. The exact causes of Alzheimer's disease is presently unknown, but at a basic level, brain proteins fail to function normally, which disrupt the work of the brain cells and triggers a series of toxic events. The neurons of the brain are damaged, they lose connections to each other, and eventually die. It's also caused by the abnormal buildup of amyloid and tau proteins in and around brain cells. Alzheimer's disease has been identified as proteopathy, and proteopathy refers to a class of diseases in which certain proteins become structurally abnormal and disrupt the function of cells, tissues, and organs of the body. It is also a protein misfolding disease due to the accumulation of abnormally folded amyloid beta protein in the brain. And the two images down below represents the amyloid beta proteopathy and taupathy in the neocortex of an Alzheimer's patient. Alzheimer's disease is also considered as a taupathy, and taupathies are heterogeneous neurodegenerative diseases characterized by the disposition of abnormal tau protein in the brain. And abnormal tau proteins are also called microtubules associated protein tau. Taupathy is a term that encompasses both the loss of function effects on the microtubules and the gain of function effects of the toxic tau species. And the image on the bottom left shows a PET scan of a normal brain and a PET scan of a person with Alzheimer's disease brain. So in healthy brains, tau proteins bind to and stabilizes the microtubules, and in Alzheimer's disease, the tau proteins come apart from these microtubules and cling to each other instead. This results in tau tangles that kill off the neurons.
Tau hypothesis. It is an abnormal aggregation of the tau proteins in neurons. Taus are regulated by phosphorylation. Abnormal chemical changes cause tau to detach from microtubules and stick to other tau molecules, forming neurofibrillary tangles which blocks the neuron transport system. Chronic inflammation. Chronic inflammation caused by the buildup of glial cells. Microglia engulfs and destroys waste and toxins in a healthy brain. TREM2 normally help fight inflammation in the brain but doesn't function normally, so plaques build up. Microglia and astrocytes collect around the neurons but fail to perform their debris clearing function. Some of the symptoms include the early symptoms: forgetfulness, repetitiveness, increasing anxiety, agitation, and period of confusion. The middle stage symptoms include increasing confusion and disorientation, obsessive repetitive or impulsive behavior, delusions, aphasia, disturbed sleep, frequent mood swing, difficulty performing spatial tasks, and hallucination. Later symptoms include dysphagia, difficulty changing position or moving around, weight loss, urinary and bowel incontinence, gradual loss of speech, short and term memory loss. In treatment there is no existing cure in the present, but there are medicines to slow it down, such as acetylcholinesterase, ACHE inhibitors, memantine, and treatments that involve therapies and activities such as cognitive rehabilitation, ruminants and life story work involves favorite possession or music, life story work, notes, and keepsake for your childhood to present day.
Keratin. Keratin is composed of fibrous structured proteins. It is an individual molecules combined to form insoluble structures. Keratins are the molecular basis for hair, nails, wool, feathers, brakes, claws, and horns that are found in mammals, reptiles, and birds. Keratin is also found in the cytoskeleton of cells, which has special function such as adhesion, migration, and metabolism. Keratin clusters are found on chromosomes 12 and 17. There are two types of molecules in keratin: alpha keratin and beta keratin. Alpha keratin is cysteine rich. It is an isomer found in cytoskeleton, while beta keratin is cysteine poor, which is an isomer found mostly in birds and reptiles. It is the building blocks of scales, feathers, and claws. It is rich in residues with small side of chains: glycine, alanine, and serine. The alpha form of keratin can be stretched up to 120% in moist heat. Beta form is rigid. This concludes that alpha form is softer than beta form. Cysteine can form disulfide bridges with other cysteine residues. These cross-linkages decrease the elasticity of alpha keratin. That means that if disulfide bonds is established with cysteine residues, the alpha keratin won't be as flexible as before. Reduction of the disulfide bonds to the sulfhydryl groups results in dissociation of the peptide chains, the molecular weight of which is 25,000 to 28,000 each. Keratin occurrence. Keratin filaments are abundant in keratinocytes. In the cumptified layer of epidermis, keratin filaments are present in epithelial cells in general, which are mostly found in our skin. The A-keratin, or alpha keratin, are found in all vertebrates. The harder beta-keratins are found only in the sauropsids, that is all living reptiles and birds. They are found in the nails, scales, and claws of reptiles, some reptile cells, testudines such as tortoise, turtle, terrapin, and in the feathers, beak, and claws of birds. Horns such as those of the impala are made of keratin covering a core of live bone.
To continue our discussion, keratin 1 or keratin 1 is a protein coding gene which provides instructions for making a protein called keratin. In the human genome, keratin genes are clustered at two different chromosomal sites. They are located on chromosomes 12 and 17. The neutral basic keratins are found on chromosomes 12, while the acidic keratins are found on chromosome 17. Let's go to alpha keratin. Alpha keratin is a polypeptide chain, typically high in alanine, leucine, arginine, and cysteine, that forms a right-handed alpha helix. Two of these polypeptide chains fuse together to form a left-handed helical structure known as coiled coil. So alpha keratin is coiled coil. It is strong, inextensible, insoluble, and chemically inert. It is found in hairs, wools, horns, and tails. Let's go to beta keratin. Beta keratin is also a member of structural protein family found in the epidermis of reptiles and birds, and it is also found in silk fibers. Beta keratins were named so because they are components of epidermal stratum corneum, rich in stacked beta pleated sheets, in contrast to the alpha keratins. So what are the importance and the main functions of keratin? Keratin is, as we have discussed, a type of protein found on epithelial cells, which live like outside surfaces of the body. So its function is to adhere cells to each other and to form a protective layer on the outside of the skin.
Functions of keratin proteins.
The role of keratin is a vital protein for the production of human skin, hair, and nails. In fact, about 95% of the makeup of hair is simply keratin. Keratin is also used in keratin treatments, which are designed to smoothen and shine hair. In a salon keratin treatment, keratin are mixed with formaldehyde and applied to the hair with a flat iron and sealing the protein into your hair. Keratin is also the protein that protects epithelial cells from damage or stress. Keratin is an important protein in the epidermis. In epithelial cells, keratin proteins inside the cell attach to proteins called desmosomes on the surface. Keratin serves important structural and protective functions, particularly in the epithelium. The skin keratin has two main functions: to hold skin cells together to form a barrier, and to form the outermost layer of our skin that protects us from the environment. Diseases caused by mutation in keratin genes include epidermolysis bullosa simplex. Epidermolysis bullosa simplex is one of a group of genetic conditions called epidermolysis bullosa that cause the skin to be very fragile and to blister easily. Blisters and areas of skin loss occur in response to minor injury or friction, such as rubbing or scratching. Ichthyosis bullosa of semen. It is a type of familial autosomal dominant ichthyosis, a rare skin disorder. It is a genetic disorder with no known cure, which is estimated to affect about 1 in 500,000 people. Epidermolytic hyperkeratosis. It is a skin disorder that is present at birth. Affected babies may have very red skin and severe blisters. Next is keratosis pharyngeis. It is a medical condition where keratin grows on the surface of the pharynx, that is the part of the throat at the back of the mouth. Keratin is a protein that normally occurs as the main component of hair and nails. Next is steatocystoma multiplex. It is a skin disorder characterized by the development of multiple non-cancerous cysts known as steatocystomas. These growths begin in the skin sebaceous glands, which normally produce an oily substance called sebum that lubricates the skin and hair. And the last is rhabdoid cell formation in large cell lung carcinoma with rhabdoid phenotype. And the intermediate filaments in keratin is keratin proteins form filamentous polymers in a series of assembly steps beginning with dimerization. Dimers assemble into tetramers and octamers and eventually, if the current hypothesis holds, into unit-length filaments capable of annealing end-to-end into long filaments. This is our references. And that's all. Thank you for listening.